Si no hay más preguntas, a pasar a la siguiente comunicación.
The third speaker is Thumbi Ndung'U. He works at the Africa Health Research Institute in Durban, South Africa. Ndung'U is interested in understanding antiviral immune mechanism and viral adaptation in subtype infection as pathway to vaccine development. His laboratory focuses on the study on HIV exposed in infected individuals and HIV controllers in order to obtain key information for development of the new therapies and vaccines. Today he's going to talk about the strategies on HIV cure based on hyperagood HIV infection studies. Ndung'U, please.
All right, thank you very much. I would like to thank the organizers for another opportunity to present our work here. This is my second time in Madrid now in 2 weeks. So if you see me in the streets and you think I'm looking familiar, you're probably right. So I'm going to talk about hyperacute HIV infection and some of the lessons that we think we are learning about HIV cure and the difficulty of HIV cure from an hyperacute HIV infection cohort. But first of all, just to say that we are interested in, obviously, HIV prevention, and we know that HIV prevention has been very, very effective in terms of reducing the number of new cases of HIV infection, but unfortunately, the number of new cases is coming down very slowly, and we are definitely missing the target in terms of reducing the number of new HIV infections. When we think about treatment, we know that also treatment works and has improved the quality of life of people in developed and also in developing countries. But unfortunately, also many HIV-associated comorbidities, some of which are listed here, and this makes it imperative for us to look for other biomedical interventions, both to reduce new HIV infections, but also to see whether we can cure HIV infection. So we've been interested in understanding acute HIV infection in South Africa. And in South Africa, obviously, we have very high incidence and high prevalence of HIV, so we are able to identify acute HIV infections. But in particular, we were interested in identifying people with hyperacute HIV infection, which we define as being in Phoebic stage 1 or 2. That's when you can detect viremia in blood, but the individuals have not yet developed humoral immune responses. So the viral load is detectable, but the humoral immune responses are not detectable. But also in the course of identifying people with hyperacute infections, we also identify other individuals that have acute HIV infection but have already passed through the peak viremia stage. And some of the questions that we are interested in addressing are: what are the characteristics of the transmitted founder virus, the immune responses in acute HIV infection, and why they fail, and what is the impact of early treatment. And we are interested, obviously, to see whether we can either mimic the effective immune responses or develop ways to augment the ineffective immune responses, either for a vaccine or for HIV cure. So we set up this cohort in South Africa. We call it FRESH. FRESH stands for Females Rising Through Education, Support, and Health. We recruit young women between the ages of 18 and 23, and we focus on this group because we know that the incidence is very high in this particular group. We provide them with an empowerment curriculum, so they come to our clinic twice a week, and when they come to the clinic, we do some socio-economic interventions to try and see whether we can intervene to see whether we can reduce some of the factors that put people at risk of HIV infection. And we also provide PrEP, and we have written an editorial recently describing this cohort in Science Immunology. So as I said, when the individuals come to the clinic, we take a finger prick blood draw, and we can test for HIV RNA. We also take serial samples pre-infection and post-infection to enable us to study viral factors and host factors associated with acquisition and disease progression. So far, in this cohort, we have identified for 74 acute HIV infections out of 1,741 enrollees. The incidence in this cohort is very high, it's 8.3. So it's what is the normal incidence in this age group in South Africa. So despite our interventions, they don't seem to be working very well. The first 14 individuals we identified, we did not treat because we were following the South African guidelines for treatment. But once we started identifying individuals with acute HIV infection, we thought that it was morally imperative that we should introduce treatment. So we started treating them. And 80% of the individuals we have identified were in FIBIC stage 1. Most of them are within 4 days of the last HIV, the last negative HIV RNA test, and we normally start antiretroviral therapy within a day of detecting acute HIV infection. So this is the data from that we already published earlier this year in Lancet HIV, showing that when you treat acute HIV infection either with a 4-drug regimen that includes raltegravir or a 3-drug regimen that is a standard of care in South Africa, you can very quickly suppress viremia to undetectable. And for the most part, this remains undetectable, and this is obviously in contrast to the people that were not treated. And by using a 4-drug regimen that includes raltegravir, which we withdraw after 2 months after full suppression, you can very quickly suppress viremia to undetectable. We've also shown that treatment of acute HIV infection, as you would expect, blunts peak viremia. So the peak viremia is around about 10,000 copies compared to about 10 million copies for the people that were not treated. And also the NADA CD4 count is much higher, significantly higher for the people that were initiated on treatment during FIBIC stage 1 compared to individuals that were not treated. We also showed, again, in that same paper that when you treat individuals with acute HIV infection, you, particularly the FIBIC stages 1 and 2, you completely prevent seroconversion. So this is data from the first 14 individuals that were not treated. You can see that all of them become Western blot positive, as indicated by these red dots, whereas the individuals that were treated during acute HIV infection, most of them don't seroconvert, particularly when they are in FIBIC stage 1. In fact, the individuals that do seroconvert, like this individual here, are individuals that we missed during acute, during the hyperacute phase, during FIBIC stage 1 and 2. And this data is also true when you look at other seroconversion tests like rapid tests or ELISA. So we've been interested in looking at immune responses in these individuals, and I'm just going to show you some preliminary data here on innate immune responses. So we have actually used a single-cell analysis to look at what are the immune responses that are generated during acute HIV infection. And in these 4 individuals, what we did is that we sampled them during the hyperacute phase, from the time that they were HIV negative to the hyperacute phase and during the acute phase of infection. And we do, using a platform developed by Alex Shelek at the Reagan Institute and his group, we have been able to sequence, RNA sequence all the individual single cells from PBMC, and these are the data showing over 600 single genome transcriptomes that were generated from total PBMC from these individuals during acute HIV infection. And you can see that you can't really differentiate between different patients in terms of the transcriptome of different cells, but you can as well look at the expression of different genes over time, and each of the dots here again represents a single cell that was analyzed, and the colors depict the times at which the single cells were obtained from the individuals. So we've used hierarchical clustering, and you can show that you can determine the transcriptome of each single cell. And when you then reconstruct that data, we can annotate various cell types and look at gene expression patterns and look at them over time. And once again, when you do that, you are able to differentiate between different cells that you expect to see in PBMCs. So the CD4 T cells, for example, cluster together. They generally express the same kind of genes. If you look at cytotoxic T cells, again, they cluster together. You can identify proliferating T cells that are only mostly common during acute infection phase. And in some of the individuals, we are able to identify different monocytes, including what we are calling super monocytes that both express antiviral genes in high levels, but also inflammatory markers in high levels in these supermonocytes. So this is a way to really look in great detail at the different cell types. And just looking in detail at monocytes, again, you can track them over time and see what are the genes that they express, particularly during the hyperacute phase of infection. And you can differentiate between those that are more pro-inflammatory versus those that have antiviral, an antiviral gene signature. And you can look at different individuals and look at which are the genes that are highly expressed in each of the individuals and whether they correlate with viral load. So, for example, in this particular individual, there's a high expression of antiviral genes during the acute phase of infection, as shown here. And in this particular individual was then followed over time, and you can track and see whether this person becomes a controller or a progressor by looking at this data. So we think that this kind of data allows us to look in great detail at what are the innate immune responses, particularly during the hyperacute phase of infection, and hopefully be able to also differentiate between what might lead to long-term control in some of these individuals, because the untreated individuals, some of them became long-term non-progressors and have actually completely controlled viremia. But the other thing that we have been interested in is looking at CD8 T-cell immune responses. Again, we are interested in looking at CD8 T-cell responses because we know that CD8 T-cell responses play a significant role in control of viremia. And unlike humoral immune responses, where we saw that most of the individuals don't make humoral immune responses, these individuals almost invariably, almost all of them make CD8 T-cell immune responses, whether you look by ELISpot, by tetramer or CFSC proliferation. And although they make a lower magnitude of CD8 T-cell immune responses, as shown here, where the frequency of single tetramer that are HIV-specific is much lower in the treated individuals compared to the untreated, these cells seem to be more functional, again, as shown here by the fact that they are producing much more interferon gamma compared to individuals that were not treated. So we think that by early treatment, you may be preserving at least some functions of CD8 T-cell immune responses. And the same thing is also seen when you look at CD4, HIV-specific CD4 cells. You can show that when you treat in phoebic stage 1, you get very good CD4 T-cell proliferative responses, both during the early phases of infection, but also during the chronic phase of infection. In this particular individual, this individual was examined at more than 250 days post-infection. And this is in contrast to this individual who is not treated, where you see that there is a little bit of CD4 T-cell proliferation during the acute phase of infection, but HIV-specific CD4 cells are pretty much gone, and even the ones that are there are not proliferating during the chronic phase of infection. And you can show that there is a significant difference between individuals that were treated during phoebic stage 1 compared to the untreated in terms of CD8 T-cell proliferation during the acute phase, but more importantly, we think during the chronic infection phase, because we think that this could be important in terms of recalling some of these responses with a vaccine strategy if we were to intervene in this cohort with a vaccine strategy. So the other thing that we have been interested in characterizing is the nature of the transmitted founder virus in this cohort. And these are some of the broadly neutralizing antibodies that have been that are available to the field. And I have highlighted here in blue some of the ones that we have tested against the transmitted founder viruses within the fresh cohort. So these are viruses that we amplified and cloned during the hyperacute phase of infection. So all the participants were in phoebic stage 1. And you can see that in majority of cases, the virus is very homogeneous. The transmitted founder virus is very homogeneous, indicating that most of these transmissions were from a single variant of HIV. As you can see here, they are genetically homogeneous. And in red here, I've highlighted the clone that we then took from that particular individual that we tested for ability of broadly neutralizing antibodies to neutralize that virus. And I would like to highlight this particular participant here, 271, because they were infected with 2 different variants of HIV during the acute phase, and we therefore took 2 of these variants, tested them against the broadly neutralizing antibodies. So these are some of the data, and again, this is just representative data. And what we find is that there's no single antibody that neutralizes all the transmitted founder viruses, even for this small set of samples, particularly when you look at antibody concentration at 0.1 mg per ml. However, most of these, some of these antibodies do quite well in terms of neutralizing most of the viruses. In particular, I would like to highlight PGT151, PGT121, PGT1400, and CAP256, which show pretty good coverage, particularly at the 10 micrograms per mg here. And all these assays were done with the TZMBL luciferase assay. I highlighted the one patient that was infected with 2 different variants. This is 271. And again, you can see that one of these clones is very sensitive to PGDM1400, for example, and the other variant that infected this individual is completely resistant to that particular variant. And I think that this particular slide kind of underlines some of the discussions that were from Marina Kaski's presentation this morning, that using one broadly neutralizing antibody may not be good enough, either for blocking infection in prophylaxis, but also in treatment with the broadly neutralizing monoclonal antibodies. And we have done some work where we have sequenced some of these individuals that show these differences in terms of sensitivity, for example, to the V1, V2 broadly neutralizing antibodies. We are focused on these antibodies because these are antibodies that we would like to use in our cohort and in South Africa, and have been shown to have very good coverage against clade C viruses. And again, you can see that it takes just one single amino acid change, for example, between this 271A and 271B clones to actually see this profound difference in terms of the neutralization sensitivity to this broadly neutralizing monoclonal antibodies. Again, and I think emphasizing that both for prevention, but also perhaps for treatment, you're probably going to need to have more than one broadly neutralizing antibody in order for them to be effective. And again, this is emphasized by this study that by Dan Baruch and Boris Yu, where they look at PGD1400 and PGT121 individually, and you can see that these 2 antibodies individually block quite a significant number of viruses, but there's a significant gap in terms of the viruses that are not covered by this individual monoclonal antibody. And the same case applies with PGT121, but when you combine the 2, you pretty much can go to over 95% coverage of the circulating viruses. And again, even the potency is improved as shown by the shifting of this curve to the left when you combine broadly neutralizing monoclonal antibodies. Okay, so that's the nature of the transmitted founder virus and the implications for therapeutics. We have also been interested in looking at the reservoir in these individuals, and in Fibig stage 1 treated compared to untreated individuals, you can see that the viral load is very high in the untreated individuals, the peak viremia compared to the treated individuals. Now, if you take the cellular DNA and do total DNA measurement as a measure of the seeding of the reservoir, you see that there is, at the same time that you're taking the viral load measurements, there's actually no difference whatsoever in terms of the DNA load. This suggests to us that the seeding of the reservoir is happening very, very early, and by the time that we identify these individuals with acute HIV infection, there is already spreading of at least of the proviral, of the total DNA load throughout the body, such that early treatment actually does not really significantly impact that parameter. But if you look at over time in terms of the reduction of the reservoir, you do see that the early treated individuals have a significant progressive decay, although it's very, very slow, whereas the individuals that are not treated remain with the high total DNA load over time. And again, I think that this suggests to us that it might perhaps be easier to intervene with a curative strategy in these early treated individuals compared to these untreated individuals. I just wanted to emphasize that point about what is happening to the total DNA, that although antiretroviral treatment is obviously beneficial and it significantly reduces plasma viremia to undetectable by 1 year in all the individuals that we have looked at that are adherent to treatment. If you look at the total DNA reservoir, actually it's a very slow decay process. Yes, it does reduce, but it is detectable in all the patients that we have looked at at 1 year. And if you look at the fold decrease in terms of total DNA, it's much less compared to the RNA load. Again, this would not be very strange news, I think, to most people who are familiar with this, but I think, again, it emphasizes the fact that early antiretroviral treatment may not be actually significantly blunting the reservoirs in these patients. And again, we have, in some of these patients, we are able to get lymph nodes, and therefore we have looked at the amount of virus in the lymph node using immunohistochemistry techniques. So this is an individual, for example, that was treated early, has undetectable viremia, has high CD4 counts. But if you look at the lymph node, you do see that in the T cell follicles in particular that are marked here by BCL6, that's what we use to identify this T cell zone. You can see that you can readily detect P24, and it is not just P24 antigen that you detect there. It's actually also P24 DNA, as you can easily detect again by RNA scope. Suggesting that there is ongoing virus replication. It's not just the residual virus that is being trapped in the lymph node. So there was this very sobering. So when we started this study and we started treating, we thought that at some point that we may want to intervene with treatment interruption. But unfortunately, there was this very sobering study that came out from this group that showed that in Phoebic stage 1, treated individuals that were treated for more than 2 years, and then treatment was interrupted, that the virus came back in almost all these individuals. So we don't think, we are not thinking anymore that we should interrupt treatment, even in these Phoebic stage 1 treated individuals. What we are now thinking of is to intervene with ways to maybe have latency reversal, because we think that there's a lot of latent virus that either is in blood or in tissues, and we think that perhaps the antiretroviral therapy may not be penetrating into the tissues, and therefore we need to activate different immune cells. And we think that agents such as toll-like receptor agonists, which have already been shown to have potency latency reversal impact, but also to be immune modulators and able to penetrate anatomical sites, may be one way to intervene in this cohort. The other approach that we are thinking of is to intervene with broadly neutralizing antibodies. I think I'll go through this very quickly because Marina this morning kind of nicely explained it, that broadly neutralizing antibodies are expected to have direct antiviral activity against the reservoir viruses, but also against transmitted founder viruses. But they can also form immune complexes with the virus and be picked up by dendritic cells, leading to stimulation of cytotoxic T cells, and also to stimulation of innate cells such as NK cells. But also they can also generate new humoral immune responses. So we think that this might be a good way to intervene in this particular cohort. So what we are now thinking is that because we are intervening early, that we are preserving immune function by early intensified ART initiated during acute phase. We think that an intervention with a toll-like receptor agonist to reverse latency in some of the individuals that have latency, including in tissues, would be a good idea. But also we need to disrupt the sanctuary, and we are thinking of using broadly neutralizing antibodies, which will have both direct antiviral activity, but also be able to stimulate new immune responses, and hopefully this will result in HIV remission. So in conclusion, we think that acute HIV infections offer new novel insights into the characteristics of the transmitted founder virus, immune responses, and reservoir establishment. And this could be important information, obviously, for cure and vaccine strategies. And single-cell analysis combined with the traditional approaches offers the opportunity to understand how immune array responses may shape the long-term outcomes in HIV infection. In the participants that we initiated on treatment during Phoebic stage 1 and 2, there was diminution of humoral and CTL immune responses, but the CTL responses may be more functional. And in the fresh cohort, the reservoir is established very early, and even in phoebic stage 1 treated individuals, the total DNA during acute infection is very similar to untreated participants, and there's also persistence in tissues. So we think that added treatment is leading to a very slow decay of the reservoir and may not be very significantly impacting on the reservoir in the reservoir sites. And we think that combination approaches which combine added treatment plus immunomodulators and specific antiviral agents such as multiple antibodies may be more likely to result in long-term remission in these participants. And finally, I'd like to just acknowledge my co-workers and the study participants and everybody who has really contributed, and also our funders. Thank you very much.
Thanks, Arnaldo. Question, please. Juan? Juan?
Thank you for your presentation. Thank you for your presentation. My question is, do you have biopsies in lymph nodes more than 2 years after starting treatment? This is just that the lymph node takes longer to get suppressed, or
we have lymph nodes from about 50 participants now, and they range from, although they are not multiple lymph nodes from the same person, so they're from different individuals. But because there's a big time range in terms of when they were collected, we think that we are still able to look at the kinetics of how the virus decays in the lymph nodes. And our finding is that in almost all the lymph nodes that we have looked at, you can detect both P24 antigen, but also RNA in the lymph nodes, even when you can't detect anything in peripheral blood.
How long after you started?
About 4 years after treatment. Yeah, 4 years after treatment study.
Okay, thank you, Thumbi, for the presentation. I have a question. You have this decrease in the proviral load. That's which will be the explanation? I mean, there is a immune response that is working better in these particular women that control the propagation of the virus and target specifically infected cells, because there is this decay is a reality.
Yeah, we do see some association between CD8 T cell activation during the acute phase and the amount of DNA, total DNA at 1 year post-infection. So we think that those CD8 T cells that are generated early might be doing something. But we actually think that the long-term sort of control of that DNA has more to do with the antiretroviral therapy rather than the immune response, because we still see diminution of that total DNA load, even when you can no longer detect immune responses. The DNA load continues to decrease. So we think that the antiretroviral treatment is somehow contributing to the reduction in that total DNA. But initially, the CD8 T cells do seem to contribute to that drop.
Thank you. Regarding this lymph node replication in your samples, how you explain the lack of seroconversion in those patients that seems to show some level of viral replication? Are those patients that seroconvert, those that have this viral replication, or can you comment on that?
So all these patients are completely seronegative. So it's hard for me actually to explain why we do see some viral replication in these individuals. I think that it could potentially mean that, and this is just speculation on my part, that maybe the virus that is replicating in the lymph node is somehow not being exposed to the immune system that much. In other words, there could be a lot of cell-to-cell transmission that is happening in the lymph node, which then means that the immune system doesn't see that virus very well, and that means that there is then no seroconversion. But that is a speculation on my part. But I should mention that when we sequence the virus in some of those individuals in the lymph node, we do see that the virus there is different from the virus in peripheral blood. And in fact, if you sequence the GAG, it has some of the patients, they have mutations within the helix binding domain, which is the domain that the virus uses to bud from cells. So we think that there could be it could indicate that there is ongoing cell-to-cell transmission, and the virus does not bud from the infected cells. Rather, that it's more like cell-to-cell transmission. But a lot of what I've said is speculative.
So, thank you very much for your presentation. I noticed in the last part of your talk, when you talk about the interventions or potential interventions in your patients, that you escaped from using CTL-based vaccines. Is that because you don't think there is a right candidate right now, or it's because you don't believe in them, or what's the point? Because you are a CTL guy in that regard.
Oh yeah. I should definitely put them there. It's a mistake on my part not to include CD8 T-cell based vaccines. I think they should be included, but one caveat is that in South Africa, we do see a very high level of transmission of immune escape variants. So a lot of these individuals, when we look at them and see the transmitted founder virus already is an immune escape variant, and that kind of raises the question about whether even if you vaccinate these individuals, the virus in the reservoir might already be an escaped virus, and you may not have very good immune responses. But I think it's something that we really need to test and not speculate on.
Después José María y después Eloisa, ¿vale? Y ya vamos a dar.
Congratulations for your excellent talk. You explained that some of these women were taking PrEP.
Yeah.
So did you see differences in the reservoir and virological and immunological data that you have explained in those who were taking PrEP and the PrEP failed?
So I should have probably qualified my statement by saying that PrEP, we only introduced it recently because in South Africa, first of all, it's still not yet publicly available, but we decided that we should introduce it as part of our study for ethical reasons. But so we haven't had enough time for me to really comment on whether there's going to be a difference in reservoir between those who are taking PrEP and those who are not. Also, the reason that we delayed in terms of introducing PrEP, there was a lot of debate about 2-3 years ago about whether PrEP was actually working in African women. But now I think there's a lot of evidence showing that it's working. So, yeah, in summary, we don't have enough data at the moment to compare those who are on PrEP and those who are not on PrEP.
Thank you, very nice talk. I just have a comment and I would like to know your opinion. We work with broadly neutralizing antibodies and neutralization assays and these kind of studies. And we have the feeling that what we know now that our broadly neutralizing antibodies have been defined with panels of viruses that are mostly from chronic infection.
Yeah.
So we have some data that say that maybe if we use a panel of viruses that are recently infected, these broadly neutralizing antibodies wouldn't be so broad. And maybe we have to start looking for antibodies that can neutralize most of the viruses from acute infection. And I'm talking about the work from Nuria in our group, Nuria Gonzalez. So do you think that this could be the case?
I think there's a need to characterize more transmitted founder viruses. As far as I know, there's only very few studies have looked at the impact of broadly neutralizing antibodies on transmitted founder viruses. And but if you look at that data, most of those viruses were more towards the chronic phase. So I think there's a need, obviously, to look at more transmitted founder virus. And I think the The other question is whether the transmitted founder viruses might have unique qualities that are different, that might make them more resistant to broadly neutralizing antibodies. I don't think we know that question. I think it's worth exploring in more detail.
Yeah.
And of course, the clade issue, because in South Africa we have subtype C, and most of the work that has been done, for example, is on.
I have the feeling that maybe it's not that they are more resistant. Maybe they are sensitive to different monoclonal broadly neutralizing antibodies, not the same ones. But as you said, we need panels of viruses from acute infection to explore that issue.
I think we need to just test first with the broadly neutralizing antibodies that are currently available and see which ones work best against transmitted founder viruses. And it might be that the ones that work best against transmitted founder viruses are different from the ones that work best against chronic infection viruses.
Yeah, I think that on the top of that, we have to use different panels to search for new broadly neutralizing. Thank you.
Bueno, muchas gracias. Vamos a dar por cerrada esta mesa y agradeceros vuestra participación y a la organización la oportunidad de haber compartido este rato con vosotros.